Welcome to the Medical Device Made Easy podcast. Here is Munir Al-Azouzi from easymedicaldevice.com. And today we will help you to calculate your sample size. So there are a lot of places where we need to calculate the sample size. I had myself to do that when I created some PMCF documents or some working on some clinical trials. So I think it's really an important topic that people should understand. And I have with me Jon Bergsteinsson, who is the VP of Global Business Development at SmartRail in Denmark. So welcome, Jon, to the Medical Device Made Easy podcast.
Hi, Munir. Thanks for inviting me.
You're welcome. So, Jon, as I've said, you will try to help us to calculate our sample size. I know it's not maybe something easy to do for some people. We will not make some equation or calculation today. So, don't think it will be a boring thing where we will just talk about some equations, but we'll more explain to you what it is and how we should do that. But before that, Jon, so can you make an introduction of yourself so that we have a better understanding of what you are doing?
Of course. So my name is Jon Bergsteinsson and I'm the co-founder and VP of Global Business Development at SmartRail. My background is actually from Biomedical Engineering and Informatics. And for the last 10 years, I've worked within clinical research and medical devices. And throughout the years, I've held different roles, everything from clinical data management expert to a data entry and site staff member in large multi-site clinical trials. But in my current role, I'm mostly occupied with knowledge sharing and insights of clinical data collection with the MedTech industry.
So great. And you are located in?
I'm located in Denmark, but originally from Iceland, a small nation in the North Atlantic.
So you start to go down and down and down.
Yeah, going a little bit more south to get a little bit more sun.
Exactly. Okay, so thank you for that. So let's try now to help our audience to understand more what is about sample sizes. So As I've said, we talk about sample size like we know already what it is, but can you make a small summary of what is a sample size and how we can determine that?
Yeah, I think it's a good to start off on the right side of the whole process. So what is sample size? Well, one of the first practical steps in designing a clinical study or clinical investigation or any other clinical activity is to determine the number of subjects or patients to recruit. In most cases, it's impossible to conduct a clinical study that includes the total population of interest, so the whole patient population of interest. Because usually when it comes to healthcare, populations like patients are indefinite. We always have new patients coming onto the clinical healthcare systems. The population for most diseases and applicable areas where medical devices come into the treatment process are indefinite. So instead of conducting studies with the whole population, we conduct studies with a sample from the population in the hope that the information that we gather on the sample that we're studying will enable us to make conclusions or inferences about the total population.
Yeah, I can imagine that if you are starting to make some clinical trials on all the population, there will maybe some people that will not be happy of that or will not want to do that.
And I think
because it's like torturing humans.
Exactly, and I think no company in the world, none of the big pharma will ever have enough money to ever conduct something like that. So. But I mean, in principle, there are multiple ways of determining sample size, but the most common way to determine sample size is to apply statistical techniques and formulas that can allow you to calculate the appropriate size for your study. And this is what's called sample size calculation and sometimes estimation as well.
So it means that we will select so for example, let's talk about 1,000. If we have 1,000 population, 1,000 people in the population, we'll select a percentage of this population, and then we'll call that the sample size. And it's only on this percentage of the population that will execute those tests or this study.
Yeah.
And what we are hoping that the result that we get on this sample size will be reproducible in all the population. It's not like just representing this group, but all the population that we are targeting.
Yeah.
So is it a good summary?
So it's according to statistical theory, this is the appropriate way to do it if you would like to at least try to get some kind of understanding of the whole population.
Okay, so we talked about studies. So do you have some specific studies where we need to use that, or where the law is requiring us to use that?
So when it comes to, like, in general, you should always assume that requirements, or most of the regulatory bodies do require to calculate sample size in one or another way. But when it comes to any given study, you always have to start by asking yourself or ourselves, are we looking to learn something, or are we looking to test something?
Okay.
And if we're looking to learn something, it's usually when we're doing a small pilot or feasibility or some kind of a very new activity that, because we don't have any clues about how our devices work in clinical practice. If that's the case, you don't always have to calculate sample size, and it actually depends on the local legislations if it's required or not. But if you're looking to test something, like to test that your device is significant or equivalent to something, usually done in a clinical investigation or a PMCF activity of some kind, you more often have to calculate sample size or determine sample size.
Okay, so in terms of the studies that we are doing actually with medical devices, I see the clinical investigation or clinical trials. I see also the PMCF that we have to do that. Do you see anything else where maybe that we need also that?
So when it comes to PMCF, first of all, the definition of PMCF is slightly loose still, because we lack some experience within the area for now. But there are also a lot of companies that are now focusing on utilizing survey data, for example, for PMCF purposes. And we've gotten questions as a vendor about the requirements for sample size when it comes to surveys. And the usual answer is always yes. You need to determine appropriate sample size no matter what kind of activity that you're doing, whether it's a survey or clinical trial or something else. If you're going to use it for regulatory approvals or submissions of some sort, you need to have an idea of what kind of sample or what kind of population you're dealing with.
So it's really, I think, so let's say that this is really important in case you are claiming something, to have a good backup for your claims, to say that, yes, what I'm claiming is correct. We have some studies, we have some statistical results for that. In terms of regulation, as you mentioned, so as we have the new MDR that will come into force, we hope, because there is this publication that, or this proposal that was made now to extend that for 1 year. So it will maybe arrive in the 26th of May 2020, or 1 year after the 26th of May 2021. Is there a place where on the MDR, I mean, do we need, or is there a place in the MDR where we say that we need to calculate a sample size?
So yes, well, in short, yes. The MDR actually covers the clinical investigation plan in the annex, where it describes the need to document, for example, sample size as a part of your clinical investigation plan. And the same goes for a PMCF plan, which is described in Annex 15, if I remember correctly, where you also have to document your decisions or methods and processes related to PMCF activity. It doesn't specifically mention sample size, but I would, without any concern, state that you would most definitely have to determine or justify your sample size in the same way as when it comes to clinical investigation. And I think it's important to note that when it comes to clinical studies and sample size, there are mainly three factors at play here. It's the factor of cost, it's a factor of evidence precision, and compliance. And the compliance part is the reason why the MDR and the many competent authorities and ethical committees already today require you to justify your sample size. And that's because if you select a sample size or determine a sample size that is too big or too low, it will affect the cost of your study and the evidence precision. So for example, if your sample size is too low, it will actually decrease your evidence precision. That is, the likelihood of you being able to make an assumption on your evidence that can mirror the whole population will be decreased. And if you have a too large sample size, you will, of course, increase cost as well, but it will also raise ethical questions. Like, why have you been testing on so many if you only had to test on hundreds of people, for example?
Yeah, as we said, the ethical committees are also here to question all this and to be sure that we are treating patients correctly, and having more patients means that we are, if I can say, as I'm using this word, but torturing more patients, which is maybe not something that the ethical committee will accept. So you have really to have a good justification, for that.
And this, I mean, it also relates to high-risk devices that are maybe thought to be beneficial for very ill patients. Then it's not so much about torturing them, but more about you decided only to treat 100, whereas you could have treated 200, for example, or you only decided to treat 100 and you decided to let 100 other people in the control group get sick and die.
Yeah, true.
So these kind of ethical concerns are also up for question when it comes to the sample size.
No, it's clear.
And I think that's why the MDR is so clear in it when it comes to clinical investigation. Like this is one of the things that you just have to be able to justify.
Yeah, and as we say that the MDR requires that, so it means that the notified bodies and authorities will look for it on your studies, will look for it on your documentation. Is there what are they exactly looking for? Are they looking for the formula? Are they looking for what is exactly the hints or the things that our audience should understand that they should have on their documentation for satisfying the notified bodies?
In my opinion, I have not been through a notified body audit within a medical device company, and I don't believe I will, but they will be looking if you to comply with what is described in the MDR, of course. What's described in the MDR is that you have to justify or document your decision making. And it basically means that you've justified how you've come up to the number 100, for example, for your clinical investigation. And the process of clarifying or justifying that can be done in multiple ways, but the most common way is to use statistical justification along with clinical assumptions that you have to make. So they might not necessarily be looking for the equations themselves, because I don't believe the auditors necessarily have the knowledge for that, but they would be looking for the scientific reasonings and the statistical justifications that combine the justification that you need to determine your sample size.
No, it's clear because, as you mentioned, there are, it's really technical. I saw some of the equations that we need for calculation of the sample size, and there are a lot of parameters, and you have to calibrate those parameters to use them correctly and to define why you use them, use 10% instead of 20 or 15, etc. So this is really a lot of techniques related to that. Just to answer this question, but as it's really technique, if I am a small company and I don't have this knowledge, is it needed specifically to have a biostatistician? Or if I have somebody on my team that is really knowledgeable in statistics, is it sufficient? Or what is exactly the requirements for, or your opinion or your advice to the manufacturers that need to do this calculation?
So in my opinion, you need to have if you're using statistical methods to justify your sample size calculation, you need to have some kind of a statistical knowledge, of course, on how to use it. This does not mean that you have to have a biostatistician in-house or you have to hire a biostatistician necessarily. If you have the necessary background, for example, if you did some kind of biostatistics in other past roles or maybe during your education, that might be enough to you being able to justify your sample size. That's perfectly fine. But most clinical teams, they don't have the time to spend on the justification. So they maybe instead hire an external biostatistician or maybe have an internal statistician in-house that maybe works within other departments or that can assist with the role of or the job of calculate the sample size.
No, I think
so I don't think there's one way to do it, but like you said, many of the equations can be quite complex. And if you have a very complex device and you have complex combination of study endpoints, this can be quite easily become very high level, and this can take a couple of days for a normal biostatistician to work out on. So if you don't have that time, most preferably you would want to outsource that somehow.
Yeah. So from my side, so I'm a consultant and I'm working with some companies, and as soon as we arrive to this question of the sample size, they are thinking that I'm not knowledgeable enough to do that.
Yeah.
And I'm always saying, yes, I'm a biomedical engineer. I have made some statistics, but knowing the formula is not enough. What are the other factors that I should consider when I'm trying to do this sample sizing?
That's a very good question, because it also relates a little bit to the fact that you cannot hire a consultant just to do everything for you. When it comes to sample size calculation, just the statistical justification, the statistical part of the whole thing, which a biostatistician or statistician in general is very good at, is not enough. You have to take into the clinical factors and the clinical assumptions as well. And the only people that can deliver those assumptions are the medical device manufacturer and usually the key opinion leaders of that company. So for example, if you're trying to justify your sample size and you write something like the sample size calculation was done at an alpha of 5% and power of 80%, and we found out that we needed 100 subjects, it's not enough, because it only takes into account some of the statistical justifications that you made, assumptions that you made. But what many fail to realize is that the combination of statistical and clinical assumptions that determines the sample size applicability. So if you leave out either of the two, you're only providing partial justification. So if you monitor was supposed to assist somebody on calculating the sample size, yeah, you could maybe find the appropriate equation for the study or the test, the biostatistics that they want to do on the results. But when it comes to the fact of determining some of the inputs that are required, like what is the minimum clinical effect I would like to illustrate, you maybe don't have a clue, because you don't have the clinical knowledge and the background to do so.
No, I think it's exactly that, and I'm, as I've said, I'm always struggling to explain that to my customers, to say I am knowledgeable in statistics, but I have no clue about the performance of your products. I have no clue if it's 50% or 20%. It's you who should tell me, and then maybe we can work together. I think it's really a teamwork with the manufacturer also to understand that and to help them really to go on the right direction, to get the same result. Because when you are making those sample sizing, you are also using that to make a comparison between maybe products that are already on the market, to see if you are equivalent to them or if you are superior to them, etc. So you have really to have those knowledge to be able to be efficient.
Yeah, and I mean, one of my good friends, a biostatistician, he told me the other day that when a medical device company approaches me and asks me to calculate a sample size, the first thing I'd ask them is, what kind of study are you conducting? Because that's one of the factors that determines how you calculate sample size. And then there are like 5 other basic inputs that you have to determine. And if you as a manufacturer have not decided or determined these inputs, your biostatistician won't be able to help you. So in order to know which of these inputs you need to determine before you approach the biostatistician and what other factors you need to take into account, you need to understand the process. You need to understand the whole concept of sample size. And this is where I think most medical device companies are left a little bit varied. They don't know, they fall hands. They don't know what to do, which is why this topic is so important.
No, I think it's clear, and I hope with this episode they will really understand how to do that and what they should consider when they are doing that. So maybe a question for you. So maybe the answer is no, but is there any tool that people can use, or people can buy, or like an Excel tool or something that can people buy and just put some numbers there and the result? Or
so there are, of course, the most common statistical tests that are made in clinical studies are available within the most common statistical tools or programs that people use. And these tools, they offer you the possibility of calculating sample size using these tests, because the way you calculate sample size is you use the statistical test and then change the equation to calculate your sample size from it. So yes, there are a lot of tools, but if you give a baby a knife and a fork and they don't know how to use it, they're not going to be able to eat their food. It's the same way with a tool like that. If you deliver a tool like that to somebody, they won't be able to calculate the sample size without knowing how to move around and how to pick up the vegetables and choose what attributes they would like to include. So it's more towards the fact, again, that people need to understand the process and learn the process before they're able to use the tools for it. Because the tools are basically just allowing you to pick in the numbers and then they do the calculation instead of you having to use a calculator for it.
Okay, so I think I think we have really here a lot of the hints or the tips that people have to do. But if I am a company that is now starting a study or a PMCF or anything, so what do you recommend me to do? Where should I start? What should I do first?
So of course, you should share this podcast with everybody that you know, so you get a little bit of insight into it. Secondly, I recommend that they probably get a hold of our latest ebook, which we'll mention a little later. But in principle, it all relates back to the fact that they need to start by looking into how does the process of sample size calculation work. You need to understand that process before you start going on to calculating your sample size. And the process for sample size calculation is actually not that complex if it's broken down into a couple of segments. There are a couple of decisions that you have to make as a manufacturer when you're designing your study. This comes down to the fact of what you're testing or what you're learning if you're doing a clinical study or just a feasibility study. What kind of design are you going to be implementing? Are you going to be showing inferiority or superiority or equivalence? So all of these design attributes you have to make, and then you have to understand what are the statistical and clinical assumptions that I have to define, and then you can go about looking into how you want to calculate it. So to do right now, if you're about to initiate or start by designing your first PMCF plan or a clinical investigational plan for a new study for a device, start by trying to understand the process. And apart from the fact that we've just released the ebook that clarifies some of it, and we're recording this podcast now that hopefully will help someone, there are multiple articles and content available online that allows people to understand it a little bit better.
No, I think it's clear. And I had the chance to download this ebook. I read it completely. I can't remember how many pages, but I read it completely. And it's really a great summary, a great tool. I really recommend it. I will put that on the show notes so that people can download it. But Great. If you read this ebook, you get most of what we talked today. You get also some examples of formulas. I can't remember if there is 3 or 5 or
there are five very basic examples.
Yeah, it's very basic, but it's enough. I think as I'm calling this my company Easy Medical Device, I like when it's easy. I understand clearly, so there is no need of being a biostatistician to really understand that. But and there is at the end a checklist to say how to. I mean, what you define today. So, what should people start to do? Is it for investigational study or is it for pilot, etc.? Mm-hmm. And you are going through this checklist so that it can help you also to define how to do your study.
Yeah, exactly.
So just now, so I think now, people really understand what is sample size and how they should do that. And as we said, they should be really careful to not take into account only the statistical part of the sample size, but also the clinical part of the products, how the product should behave and what are the performance that it should achieve. so now you as Smart Trail, so what are you doing exactly? What is your company doing, and how it can help the medical device community?
Yeah, well, that's a very good question. So we at Smart Trail, we feel that every MedTech company should be able to collect high quality clinical data. So no matter the size or the budget, and it's our mission to make it easy and cost effective to collect clinical data in compliance with the regulations. So one of the ways that we do this to create software and services like SmartTrial, which simplify the whole data collection process. And SmartTrial is our leading product, and it's a do-it-yourself electronic data capture for clinical investigations and PMCF activities, such as surveys and registries. And a big part of our services is directed towards content generation and knowledge sharing within the MedTech industry, which is why one of our latest publications on the medical device sample size cookbook was curated to help clinical teams to better understand the process behind sample size calculation, in the hope that it will make it easier to comply with the requirements set forth by the MDR, but also by other regulatory bodies like the FDA and so forth. So yeah, I suggest that everybody take a look at it, download it from our website or from the link that you'll provide with the podcast.
Yeah, I will put that on the show. So if I understand well, you have your application or software or platform that is connecting, if I can say, the hospitals with the data, I mean, collecting the data from hospitals or from anywhere, and then you are processing them or you are just storing them?
So we have medical device companies are using SmartTrial as a data collection tool for their studies. So they're implementing it in sites that are working with their devices, where they have physicians entering data directly into SmartTrial during the course of a clinical study or a PMCF activity of some sort. So the data can be directly entered into SmartTrial on a tablet or a computer on site, and then the data is kept securely in the SmartTrial system and accessible by the sponsor if needed, which can then export the data for further analysis and then reporting for MDR purposes, for example.
So before SmartTrial, how people were doing that?
So the MedTech industry has actually been quite far behind when you compare it to other industries. Pharmaceutical companies have been using software like SmartTrial for years, but most MedTech companies have actually been using paper and Excel to date.
Because I remember that only when I received some of the data that I had to use to create the PMCF or the clinical evaluation report or studies, etc. Yeah. I really received some papers filled by people, and I had to collect that and to compile that, which is painful. So it's good to have a software like that that can really collect everything, and I have just maybe to apply some formula or anything to make my data calculation, which is great.
Exactly.
Okay, so great. So thank you for that, Jon. So in terms of the people that are listening, so yeah, as I've said, I will put everything on the show notes. I will put also the details of Jon and the SmartTrial on the show notes. So if you want to go and check that. As I've said, really, this ebook is really complete. I mean, complete for people that want really to understand what is a sample size. There are some formulas, so you can try to use them and see if you arrive to the right numbers. But yeah, it's really, I think, an important topic. As we talked today, don't make it like it's easy just by taking a formula and putting some numbers. You have really to have you have to understand the process and understand your product so that you can really reach the right objective. And then notified bodies will be happy for that. So I think this is the most important thing, is that at the end, when the notified body is reading your report, they are not having any question on saying, why did you choose 100 or 56 or this or that on your product?
Exactly.
Okay, so Jon, so where people can follow up with you? So I think LinkedIn is one of your platform.
LinkedIn is probably one of the primary platforms for myself. So you're welcome to follow me if you can find me there, Jon Bergsteinsson. I'm also fairly active in our LinkedIn group, which is the only community for clinical teams within the MedTech industry.
Okay.
It's called Clinical Investigations and PMCF for Medical Devices. So I'd be happy to have you join us there as well. Otherwise, I'm also a little bit active on our blog. So
So I will put anywhere all the links so that people can follow up with you and contact you maybe to ask you a question, maybe to help them to understand the sample sizing process.
Okay. Happy to at least to guide them into the right direction.
No problem. So thank you for that. Okay, so thank you, Jon, for your help, and I think really this was a really helpful topic for the people. For the people that are listening, so don't forget also to share that with people that are really trying to understand what is sample size. Also to people that want to learn more about medical device regulation, medical device standards, so how to apply them. I'm really trying to educate people about that. And if you are looking at that through YouTube, don't forget to like and comment and provide me also your questions if you have any questions. I will transfer them to Jon if there is anything that I cannot answer by myself. Okay, so thank you, Jon, and I wish you a nice day.
Thanks for having me, Monier. Have a good day as well.
Bye-bye.